DNA sequences code for specific proteins. This strand here is the coding strand of the DNA, which means that it is the exact same as our mRNA, but the thymines or T's become uracils or U's. The template strand is used to replicate the mRNA, but because both the coding strand and the mRNA are complementary to the template strand, we can use the coding strand for ease of translation. We will be using this strand to illustrate the different types of single nucleic acid or point mutations. These changes are most prevalent at the amino acid stage, so for the example strand, we will go through the steps of translating and transcribing it to get to a point of reference for the others. Because this is the coding strand, like I said, all we must do is replace the T's with U's. The adenine or A, cytosine or C, and guanine or G all stay the same. Once this is done, we look for our reading frame. Each amino acid is made up of 3 codons, but they all start with a specific amino acid called M. This amino acid codon is AUG, so we must find AUG to figure out where our frame starts. AUG is right here, and that is where our sets of 3 begin. I'm going to use the one-letter abbreviations for the amino acids. Going from there, we get W, N, A, C, and finally a UGA codon, which codes for a stop. This doesn't have an amino acid. What it is actually doing is telling the enzyme that is transcripting to let go, that this is the end of the protein. First, we will start with a silent mutation. All these changes happen in the original DNA, are now translated and transcribed. I'm going to change the C in the third set to a T, and then translate and transcribe. The reason that it is called silent is because although it is a different codon in the DNA, it still transcribes to the same amino acid in. This mutation has no effect on the protein created. This change from a C to a T is called a transition. This is because C and T are both pyrimidines, and when a base changes to one of the same types, it is called a transition. This can also be done between A and G because those are both purines. Next, we will go over a missense mutation. I'm going to change that same C from last time, but this time into a G. Transcribing changes the entire amino acid into a K. Missense is when the change results in a different codon and a different amino acid. This change of a C to a G is called a transversion because C is a pyrimidine and G is a purine, which are 2 different types of bases. This mutation will influence the protein, but the amount may vary. Next is a nonsense mutation. I'm going to change the first G and the second codon into an A. This causes the codon to now be UAG, which is a stop codon. Nonsense mutations are when protein formation stops earlier than normal. This change from a G to an A is called a transition because they are both purines. This would mess up the formation of the entire protein. Lastly, we have a read-through mutation. In the third to last codon, I'm going to change the G into a T. This was our stop codon, but it now codes for L, which means that now the codons after this will get transcribed too. It is called a read-through because protein formation extends longer than usual. This change from a G to a T is called a transversion because G is a purine and T is a pyrimidine, which is 2 different types of bases. This would most likely mess up the formation of the entire protein.